In 2 weeks, scientists working off Brazil confirmed 31 deep ocean species that science had never documented before. Not 31 animals. 31 species. They included 9 jellyfish, 7 colonies that behave like single animals, a transparent worm built for unexpected speed, and 2 organisms so large that it is hard to believe each one is only a single cell. The expedition team confirmed them as new. Formally describing and naming each one will take longer. The speed sounds impossible because identifying a new species can take years. Part of the answer was the technology on the ship: lasers, 3-dimensional imaging, live microscopy, and DNA sequencing, all in one place. But the real breakthrough was not simply finding the animals faster, it was seeing them before collection changed them. A jelly could enter a net with its tentacles and body intact, then reach the deck as a transparent mass, missing the features needed to identify it. The discoveries came from the midwater of the tropical South Atlantic, the enormous space between the bright surface and the seafloor. It looks empty from above, but it is filled with animals made from transparent tissue, mucus, and water. In the deep ocean, finding an animal and truly seeing it are not the same achievement. And the reason so many remained difficult to identify becomes visible the moment one enters a traditional net. A jellyfish can be alive, structurally complex, and almost impossible to bring home intact. Traditional ocean sampling often depends on nets. A net can cover a large volume of water and recover animals from depths no diver could reach. It remains an essential research tool. But imagine what happens when a body made mostly of water meets mesh, turbulence, pressure change, and a long trip to the surface. Tentacles break. Transparent tissues collapse. A colony arrives as fragments. The specimen in the jar may preserve enough material for genetics, yet lose the body plan that distinguished it from its closest relatives. That creates a strange scientific blind spot. Hard-bodied animals leave clear specimens. Delicate midwater animals may leave something closer to biological wreckage. The problem is made worse by the scale of the habitat. In the midwater, there is no reef or seafloor to concentrate life. An animal can drift above, below, or beside the rub and disappear without ever entering its lights. Even when a camera finds one, a flat image may hide the feature that matters. A jelly can rotate, a siphonophore can contract. Transparent tissue lets one structure overlap another. A biologist trying to identify the animal later may be looking at one angle of a shape that changed before the next frame. This is why the 2026 expedition off Brazil did not rely on a single way of seeing. The remotely operated vehicle, Sebastian, carried several imaging systems into the animal's own environment. Instead of forcing every discovery through a net, the team could examine some creatures while they were still suspended in deep water. The first shift was simple to describe and difficult to build. Do not flatten the animal into a photograph. Scan the space it occupies. Deep PIV passes laser light through a transparent animal and reconstructs its 3-dimensional shape along with the water moving around it. Iris uses multiple viewing angles to create precise 3D images in real time. Then a shadowgraph camera reveals finer internal and external structures that the other scans may miss. Each one answered a question a flat photograph could not. Which apparent line is an organ, and which is simply another transparent surface behind it? This did not make collection obsolete. It made collection more intelligent. Some animals could now be documented in detail without immediate collection, while selected specimens could still provide tissue and genetic evidence. Even if a body collapsed later, the scan preserved the shape and position of structures that would otherwise disappear. And once scientists could preserve the animal's shape, the expedition exposed a second problem. A strange shape alone does not prove a new species. the team still had to show that these creatures were genuinely different, not damaged examples, juveniles, or unfamiliar life stages of something already known.
It's definitive.
The 31 species did not look like variations of one animal. One was a colony whose members divided the work of swimming, feeding, and reproduction. Another was one giant cell. A third lived inside a mucus filter larger than its own body. The discoveries included jellyfish, siphonophores, comb jellies, larvaceans, giant single-celled rosarians, an amphipod, and a gossamer worm. But a few examples reveal just how different these species were. Consider the siphonophore. From a distance, it can look like one long, luminous animal. Up close, it is a colony formed from many specialized zooids. Some parts propel it, some capture food, some digest, some reproduce. The colony moves as one body, even though no single zooid performs every job. Now compare that with a rosarian. What looks large enough to be a many-celled animal can be one enormous cell, often supported by an intricate mineral skeleton. The scale of the body tells you almost nothing about the number of cells inside it. Then there are the larvaceans. Their small bodies resemble tadpoles, but the structure around them may be far larger than the animal itself. They secrete mucus houses that filter particles from the water. When a filter clogs, the animal abandons it and builds another. The discarded house sinks, carrying trapped organic material downward. The gossamer worm challenges a different assumption. Its body appears fragile and ribbon-like, the sort of form a viewer might expect to drift passively. Yet the expedition documented one moving faster than its shape led scientists to expect. That gap between appearance and performance is exactly what in-place observation can reveal. A preserved specimen can retain parts of the anatomy. It cannot preserve the sudden burst of acceleration recorded while the animal was alive. Every group demanded a different kind of proof. Jelly anatomy can be subtle. Siphonophore colonies can be mistaken for fragments. Comb jellies are fragile and can alter shape. Larvaceans carry structures that disappear during collection. Single-celled rosarians force the observer to stop equating visible size with multicellular complexity. That created a scientific risk. When everything looks unfamiliar, novelty can make almost anything appear new. Scientists could not simply point to an unfamiliar animal and announce a species. They needed comparisons with known specimens, specialists who understood each group, clear morphological differences, and genetic evidence where suitable. The expedition team reported that experts were able to confirm 31 species as new. New to science is not a dramatic synonym for we have never personally seen this. It is a testable claim that the organism differs from species already documented. The ship had found a way to accelerate that test. Seeing the animal alive was only the first half of that test. The second began inside the ship. A deep-sea specimen starts changing the moment it leaves its habitat. Temperature rises. Pressure falls. Tissues relax or rupture. Pigments fade. A body that remained transparent in the water may become cloudy. If the animal is tiny, a single damaged feature can erase the difference between one species and another. Normally, samples may travel from a ship to laboratories on land before specialists can run extensive genetic comparisons. On this expedition, the team brought more of that process to sea. High-resolution images gathered at depth were paired with genetic sequencing from collected material aboard Falkor. This allowed researchers to compare what the animal looked like with what its DNA indicated while the cruise was still underway. Instead of treating morphology and genetics as two investigations separated by months, the team could use one to challenge the other. Two jellies may look identical, but carry different DNA. Two others may look different only because one is young, damaged, or contracted. DNA can reveal a hidden lineage, while living anatomy shows scientists which visible features to look for. Neither clue is reliable enough on its own. Because the analysis happened aboard the ship, a missing feature could be recognized before the expedition ended rather than months later on land. That shortened the delay between observing an animal and making the first anatomical and genetic comparisons. In a place where every dive costs time and every encounter may be unrepeatable, that change matters. Rov Sebastian descended with cameras and instruments while scientists watched from the ship. When something unusual entered the lights, specialists from different fields aboard the ship could compare what they were seeing immediately. A gelatinous body that might appear meaningless to one observer could be recognized as important by another. The speed of the discovery came from this chain: locate the animal, document its living form, collect selectively, image finer structures, sequence genetic material, and compare the evidence while the expedition was still active. It did not eliminate scientific caution. It moved the first comparisons from months later to the same week. If the DNA appears new, but the body is too damaged to compare, is that enough evidence to call it a new species? And one instrument pushed the expedition to an even smaller scale. One of the most revealing discoveries was not a new animal at all. It was a new view inside a living cell. Inside the ship, scientists placed a large, single-celled organism under a microscope and watched its living machinery move against a glass skeleton. The organism was a protist, a single cell supported by a glass-like mineral frame. The phrase single-celled invites the wrong mental image. We tend to picture something so small and simple that only a microscope could make it interesting. Some oceanic protists destroy that picture. A single cell can form a body visible to the naked eye. Internal structures move, exchange material, and interact with a mineral framework while the organism lives. Researchers used an open-source confocal microscope called SQUID, developed through Stanford's Prakash Lab, to image living internal cellular structures in 3 dimensions. According to the expedition report, this was a first for research conducted at sea. The timing mattered. A fixed or dead cell preserves a moment. A living 3-dimensional image shows activity. The team could watch cellular structures interact with the organism's glass skeleton, rather than infer the relationship from a collapsed specimen later. That creates a scale reversal. The expedition began with an ROV large enough to require a research vessel. It descended through hundreds of meters of water to find animals whose bodies could easily deform during collection. Then, inside the ship, another instrument followed movements within one cell. The ocean's largest habitat was being examined from the size of a drifting colony down to living cellular architecture. The microscope revealed something a species name alone could never show: how the living cell functioned. If the only output were a list of 31 species, the expedition would still be important. But observing the cell alive widened the investigation. Scientists were no longer asking only what the organism was. They could begin examining how it functioned under cold, pressure, and darkness. The same question applies at every scale. The siphonophore splits the work of staying alive across a colony. The larvation builds its filtering structure outside its body. The protist organizes a visible skeleton within a single cell. Together, feeding, waste, discarded structures, and death move material through the water column. At 800 meters, the cameras captured one link in that chain: a predator feeding on a jellyfish. At roughly 800 meters, cameras recorded a female pelagic octopus, Haliphron atlanticus, feeding on a bright red jellyfish. The image is immediately understandable: an octopus wrapped around its prey as both drifted through black water. Its scientific value goes further. Feeding observations in the midwater are difficult because the habitat has no fixed stage. A stomach sample can reveal that one organism consumed another. It cannot always reveal whether the prey was hunted, scavenged, carried, or used in some other interaction. Direct footage places behavior back around the evidence, and that behavior is part of something larger. Gelatinous animals and the discarded mucus houses of larvaceans can carry organic material into deeper water, where it may remain for longer periods. This is why an unknown jellyfish matters even if it never receives a dramatic common name. Misidentify the jelly, and scientists may also misread what eats it, where it travels, and how much material it carries into deeper water. The expedition also collected a juvenile glass squid from 779 meters and a jelly from 1,157 meters. Both were photographed aboard the ship using a prototype multi-view camera system. These encounters came from different depths in an ecosystem that shifts hour by hour. Some organisms remain deep, others rise toward the surface at night and sink again by day. Predators follow prey. Carbon follows bodies. The animals are not distributed like objects on a seafloor. They migrate through a moving volume of water. That is how an ocean full of life can look empty when the observer arrives with the wrong tool, at the wrong depth, at the wrong time of day. There was another advantage to seeing these bodies before preservation. Behavior itself became evidence. A tentacle held at a certain angle in a jar might be damage. The same tentacle used again and again during swimming is anatomy in action. Compare two versions of the same encounter. In the first, only a crushed specimen and an unfamiliar DNA sequence reach the laboratory. In the second, scientists also have living footage, a three-dimensional scan, and a carefully collected sample. Uncertainty remains, but far less evidence has disappeared. This is why speed did not mean carelessness. It came from moving several slow steps into the same room. The ship had become an assembly line for evidence, and evidence is what it delivered. The technology could reveal the animal, but it could not remove the final scientific question: when is an unfamiliar organism truly a new species? There was still one uncomfortable question. Can scientists really report a species as new after only 2 weeks? The answer is that discovery, confirmation, description, and naming are connected stages, not one instant event. An expedition can gather strong evidence that organisms represent species not previously documented. Experts compare living anatomy, preserved specimens, existing collections, and DNA, and they may become confident that the lineages are new. Before a species receives an official name, researchers still have to identify the traits that separate it, compare it with known relatives, preserve a reference specimen, and publish the evidence. The expedition team could confidently report them as new, while the formal descriptions and scientific names still required further work. That is not a loophole. It is a record of work still in progress. The expeditions team included specialists capable of evaluating these groups. Their claim was not based on strangeness alone. Still, technology cannot guarantee perfection. Genetic databases are incomplete. Delicate animals may show different forms across life stages. A colony may resemble another under one viewing angle. Later research may revise some classifications. That is why the images, specimens, and sequences all need to remain available for comparison. The achievement was not rapid naming. It was preserving several kinds of evidence before the animals changed. The team reported seeing far more diversity and abundance than expected, but 31 represents only the organisms experts reported they could distinguish as new. Every dive still illuminated only a narrow path through an immense habitat. That changes the central question. The expedition does not mean the midwaters suddenly became easy to survey. It shows how strongly the available tools shaped what scientists were able to observe. The new tools reduce that bias, but they introduce their own limits. And Rov's lights may alter behavior. Its field of view is narrow. Laser systems need suitable angles. A fragile animal documented perfectly on video may still leave too little tissue for comparison. A specimen may provide DNA but lose the posture that made it distinctive. No single instrument solves the deep ocean. A scan preserves living shape. A specimen provides tissue. Na tests the lineage. Agreement strengthens the claim. Disagreement reveals the next question. The evidence was already spectacular. A transparent worm accelerating through darkness. A colony moving as one body. A visible organism made from one cell. And an octopus wrapped around a crimson jelly. The expedition's chief scientist described the midwater as the largest habitat on Earth. The phrase sounds abstract until the scale of the sample is considered. 2 weeks produced 31 new species from a limited route. Most of the habitat remained outside the camera frame. The unknown is not a dark patch at the bottom of a completed map. It is the map's dominant color. Scientists found 31 new deep ocean species in 2 weeks because they stopped asking one damaged specimen to answer every question. They observed living bodies in place, used laser scans to preserve three-dimensional form, used shadow imaging to reveal finer structures, collected selected specimens, and compared their anatomy with genetic evidence aboard the ship. The speed came from connecting evidence that was once separated by distance and time, and the record outlives the cruise. Years later, another scientist can reopen the scan, compare the genetic sequence, and inspect the specimen without needing the original encounter to happen again. A structure that looked minor could become the trait that separates two lineages. For decades, some of the ocean's most delicate animals reached the surface as broken clues. In 2026, more of them reached science as living systems. 31 new entries in a catalog is the smaller half of that. The larger half is a new way to meet creatures that our older tools kept changing before we could understand them. Which changes your idea of life more? A colony that acts like one animal, or a visible organism made from only one cell? Tell me which one you would send the next expedition to study, and why. The real breakthrough was not finding faster. It was damaging less evidence. The ocean did not suddenly become more diverse. Our way of looking finally became less destructive. That distinction matters for everything still waiting down there. Every animal too fragile for a net is an animal we have been counting as absent rather than unseen, and the midwater is mostly made of animals like that. Subscribe if you want to see what the deep ocean looks like when we stop breaking it on the way up. Next, we go down to the animal that survives a pressure that would collapse a steel hull